Hey, so we are going to do kind of a quick lecture today on chapter 2B, which is talking about the cell, some of the structures in the cell, and how proteins are made, because proteins are super important structures and functioning uh, molecules for cells. So cells, there are trillions of cells in your body, and they are the smallest functional units in the body. And so when we say functional units, these cells are actually doing functions that contribute to the function of the tissue, which contributes to the function of the organ, which contributes to the function of the organism. And so kind of a few basic things to review. A general cell is going to have a cell membrane. Cell membranes are referred to as the plasma membranes or the plasma lemma or cell membrane. And these plasma membranes separate what's inside the cell from what's outside the cell. And so inside the cell, talk about intracellular fluid, which is cytosol. And then outside the cell, you have extracellular fluid, which if it's surrounding cells in a tissue bed, it's interstitial fluid. So this is really important to maintain this, what's inside the cell from what's outside the cell. And we talked about this last lecture. It can mean the difference between potential energy. It can mean the difference between the cell having what it needs. And so the cell membrane serves a really, really important function. Now, what's inside the cell is called cytoplasm, and that can further be broken down into the liquid part. So the liquid is the cytosol. It contains dissolved things like amino acids, things you can't see, glucose, and so forth. If it's structural, it's going to be called organelles. So these are structures, and they may have membranes around them and they may not have membranes around them. So you get membranous or non-membranous. This is a prettier picture of the generalized animal cell, and you have the cell membrane surrounding the outside, and inside you have various structures. Today we're really going to be focused here on the nucleus, because the nucleus is where we have DNA, and DNA contains genes, and genes code for proteins. And so we're going to talk about how these proteins are formed, starting in the nucleus. The nucleus is surrounded by what is called a rough endoplasmic reticulum. It's rough because it has ribosomes attached to the outside. So these right here are ribosomes, and they give it a rough appearance. And so the rough endoplasmic reticulum has ribosomes. This is generally where the proteins that are started in the nucleus, then created in the cytosol, get finished. And some cells will have a smooth endoplasmic reticulum. It doesn't have ribosomes. And in the case of like a muscle fiber, we'd call it a sarcoplasmic reticulum, and it would contain calcium. So different cells have different reasons for smooth endoplasmic reticulum. This is just general stuff here. Golgi apparatus is another membranous kind of tubular structure where the finished proteins from the rough endoplasmic reticulum will get sent to the Golgi apparatus to then be packaged and tagged for either exocytosis, to stay in the cell, or, wait for later in a vesicle, form a lysosome, numerous things. And then the last thing really important here is the mitochondria. So this is showing you the mitochondria here. We call it the powerhouse of the cell, because this is where cellular energy is being formed. And I should say is being efficiently. You can transfer the energy in a sugar molecule, a glucose molecule, to ATP in the cytosol of the cell. without the mitochondria, but it's very inefficient. And so cellular respiration is really the efficient transfer of energy from glucose and triglycerides to ATP molecules. So those ATP molecules can then be used by the cell for kinetic energy. And so this happens in the mitochondria. And so we go over that in detail in chapter 3. So cell membrane, it's a phospholipid bilayer. And what is meant by phospholipid bilayer? Look, the cell membrane is number one function is physical isolation. But things have to be able to get into and out of the cell, so it has to be selectively permeable. So physical isolation between the intercellular fluid and the extracellular fluid, but there needs to be selective permeability. And the way it does this is you have these phosphate heads. So if this is the extracellular fluid, these phosphate heads on the outside from the intracellular fluid, these guys right here are polar molecules. I can't spell polar. And so what else is a polar molecule? Water. And so extracellular fluid is mostly water, it's polar, and so these phosphate heads get along great with water and other polar molecules like amino acids and glucose and so forth. And so again, and so these phosphate heads are great, right? But we don't want polar molecules to go through. You need it to be a barrier, right? You do not want things to travel through. So now the inside these lipid tails orange, are nonpolar. And so this is nonpolar lipids. And so water cannot pass through there, but lipids are flexible, right? And so that, along with cholesterol, which is lipids, make this flexible. So you can insert channel proteins to allow facilitated diffusion of substances when the cell selects to. It can insert into the cell membrane and let polar molecules into the cell or out of the cell. Gases can diffuse through this lipid bilayer, but polar molecules cannot. And so this phospholipid bilayer is really important. And that's the regulation of exchange. You can have receptors on the outside of the cell membrane or the inside of the cell membrane, and it can be structural support. So when we talk about hydrophilic, this is water loving, right? And so these are polar molecules. And hydrophobic is fear of, and so these are nonpolar molecules. So understand hydrophobic, hydrophilic, polar, nonpolar, structure of a phospholipid bilayer, and what can go through and what can't. Again, cholesterol adds to the fluidity. An example I talked about, you eat a jamba juice. This is going to increase the concentration of glucose in your blood and in your extracellular fluid, because if it's high in your blood and low in your extracellular fluid, it's traveling into the extracellular fluid. That's going to cause insulin to be released. And this insulin molecule is going to bind to a receptor on the outside of the membrane. That's going to trigger GLUT4 protein channels to then be inserted. So we're going to call this one a GLUT4. And this channel protein gets inserted, and that then allows glucose to go from a high in the interstitial fluid into the cell, which is lower, and facilitates diffusion. So you can have proteins on the outside like receptors. You can have these kind of amphipathic molecules that have this lipid portion that's okay with the lipid tails, but have kind of the polar sides of it, and it can integrate. It's an integral protein right here through the cell membrane. Peripheral protein, an example of that would be the receptor on the outside or some kind of cytoskeleton. So go through integral proteins, peripheral proteins. Again, they're ambipathic. We talked about that. There's a polar side and non-polar side. And it's giving you other examples. Enzymes, receptors can be on one side of the cell, the phospholipid bilayer, or the other. That would be a peripheral protein. Glycocalyx, you can have the outside of the phospholipid bilayer have glycoproteins or glyco structures that will bind the cells next to one another or provide, that would be cell adhesion or some recognition. It could be a protective layer. It's just kind of an FYI. Functions of the cytosol, this is intracellular fluid. This is where translation of proteins occurs. This is where glycolysis occurs. This is where you store glucose and triglycerides and secretory vesicles. So things are kind of happening or being stored in the cytosol. And then we get to the organelles. So membranous organelles. We'll start with the nucleus. Now the nucleus is important because here is DNA. And what is DNA? Well, it's the big book of proteins. So how exactly, which amino acids need to go next to which amino acids to produce protein A? That would be one gene. And so the DNA is really all of these genes that explain how to build all the different proteins, and that's stored inside your nucleus. Inside the nucleus, deep, is the nucleolus. This is where ribosomal RNAs are formed, and then ribosomal RNAs will leave the nuclear pores and go out and help build proteins. So ribosomal RNAs, we just call them ribosomes, and they help build proteins, right? So they're building proteins. Messenger RNAs get created here off a strand of the DNA, and that will also leave to go build a protein. And we'll talk about that with transcription and translation of proteins. So go through the nucleus. The nuclear envelope is really two membranes, phospholipid membranes, and there are these little holes called nuclear pores in which the RNAs and the mRNAs can leave the nucleus and go out into the cytosol. So again, nucleolus forms the ribosomes that we'll talk about later that help translate the messenger RNAs into a protein. Endoplasmic reticulum surrounds the nucleus, and this is where the finishing of proteins can occur. The ribosomes will attach to the outside and shove the protein inside the endoplasmic reticulum to finish its conformation, and then package it and send it to the Golgi apparatus. So this is right here is the Golgi apparatus, this green thing. Types, you have smooth and rough, and this goes through the, really what smooth and rough are for. Please read through these descriptions. And so you can pause me and read through those. Golgi apparatus, this is really, it's called post-translation processing of proteins. And what that really is reading what the protein is for and then tagging it or packaging it to either leave the cell, stay in the cell, wait. So example of insulin. If this was a beta cell in the pancreas, the protein being formed would be insulin. The Golgi apparatus would read that the insulin is going to then be put into these vesicles and stay and wait for the receptor to let the cell know when glucose goes up. That will then trigger the excitation of insulin inside these vesicles. So that's just an example of post-translation. So again, it's good to pause right now and read through the Golgi apparatus, and kind of this slide. It's really, I like the slide as it goes through proteins, and how they're modified, packaged, and how they travel in the cell. Moving on to the mitochondria. The mitochondria is a double membranous structure as well. There's this intramembranous space where the electron transport chain is happening, and cellular respiration is occurring. So we're going to talk in detail about the mitochondria when we talk about cellular respiration. This is where we efficiently transfer energy and mass produce ATP molecules so the cell can use to move things, or use it as kinetic energy. The mitochondria is where oxygen is consumed. So you breathe in oxygen, you have this whole respiratory tract that gets oxygen into your blood, and your circulatory system sends that oxygen in your blood to all the cells in your body. And then your cells get this oxygen, and the oxygen is going to go to the mitochondria to accept the electrons at the end of the electron transport chain and bind with protons coming out of the intramembranous space, and it will form water. Now, if you don't have oxygen available to do this, the mitochondria can no longer mass produce ATP molecules, and that becomes a problem. So this is called aerobic respiration because you need oxygen to make these ATP molecules. And we will talk about that in chapter 3. By the way, 100% of the DNA that produces or creates the mitochondria in your body is from your mother. So you have your mama's mitochondria. Lysosomes, they're just little enzymes or proteins that are produced, transcription and translation in the nucleus, and then packaged specifically to break down pathogens, viruses during phagocytosis, or debris, to break it down so then it can be exocytosed out of the cell, and the cell can get rid of its waste. Peroxisomes have they're called peroxisomes because it's kind of a hydrogen peroxide type of substance that it breaks, sorry, I said this wrong. It's peroxide, so it can break down the toxic hydrogen peroxide that can form in a cell and disable that and get rid of it. Non-membranous organelles, ribosomes, we'll talk about when we talk about proteins. Still don't quite know what those are for specifically. And then centrioles are for mitosis, when you're splitting the cell in two and forming a new cell, and the cytoskeleton is kind of the structure of the cell. So again, ribosomes, they're messenger, or sorry, they're RNA from the nucleolus of the cell and the nucleus, and they are responsible for helping build proteins. Kind of go through these and the different cytoskeleton filaments, and you can pause and read through this. And microvilli, again, these are protein cytoskeleton structures that increase the surface area of the cell membrane. If it's a cell that requires a lot of absorption, it's going to just increase the surface area for maximal absorption across the cell membrane. Cell adhesion molecules are also protein structures that will bind cells together or allow ions to flow from one cell to another, and aid in direct or indirect communication. So these cell adhesion molecules. All right, last concept we're going to talk about is the building of proteins. And so proteins, we use proteins in all physiological processes, whether it's cell signaling, whether it's rebuilding structures like actin and myosin, or muscle fiber, myofibrils, or enzymes for cellular respiration to transfer energy from the food you ate into ATP molecules. Hormones are proteins, neurotransmitters are proteins. And so we need these proteins to drive all our physiological processes. So that is to say, the DNA, which is the recipe book for all of these various proteins, is very important. And during mitosis, when you're forming a new cell, the DNA has to be copied exactly, and each new cell has a copy of that DNA. And if anything goes wrong during the copying of the DNA, you could end up with the gene not coding for the correct amino acid in that protein. And then that can then compromise the structure of the protein. And that can then compromise the function of the protein, because the function of the protein is really based on the structure of the protein. And the structure of the protein is based on which amino acid is put next to which amino acid, and that is directed by the genes in the DNA strands. So really important that the DNA codes for the correct amino acids and synthesizes proteins correctly. So DNA is in the nucleus of the cell, and this is where transcription occurs. And what transcription is unwinding the DNA strand to that area where there is the gene to code for a protein, and reading that gene and forming a structure called a messenger RNA, which is a copy of the order of amino acids. So a messenger RNA is a copy of the gene. It's a copy of the order of which amino acid needs to go next to which amino acid. The messenger RNA is created off the DNA strand in the nucleus. It leaves via the nuclear pore into the cytosol. And the cytosol is where translation occurs. And what translation is reading the messenger RNA and assembling the amino acid strand. And that's the primary protein structure that we talked about last lecture, to form ultimately a protein. So protein synthesis, this kind of showing you the DNA. Well, from the DNA strand, off a particular gene, you form a messenger RNA. That messenger RNA, and that's called transcription. That messenger RNA leaves into the cytosol, well, cytoplasm, I guess the organelles are there as well. And translation is going to occur via rRNA and tRNA to assemble the amino acids into a primary protein structure, which will then fold and be finished in the rough endoplasmic reticulum and so forth. So please know these definitions, right? DNA, what is transcription? What is translation? Where are they occurring? And the flow of genetic material. From a gene on the DNA strand, an mRNA or messenger RNA strand is produced in the nucleus. It then leaves the nucleus into the cytoplasm and is read by the ribosomes or the ribosomal RNAs, and with the help of the transfer RNAs, builds a primary protein structure. So again, a gene is a portion of the DNA holding the genetic code for a specific protein. Genome is all of the genes for all of the proteins in your body. Now, a gene, this is showing you like, gene number 1. A triplet on the DNA strand codes for one amino acid. And the next triplet will code for amino acid 2, and the next triplet will code for amino acid 3. And they're all different, and they're based in these, nucleotides. And you don't need to know GTCA for the triplet and CAGU for the codon. What you do need to understand is a triplet codes for one amino acid. And as that DNA strand is unwind, an RNA codon is formed off the triplet. So this codon is going to code for amino acid 1. And then a second codon off the second triplet is going to code for amino acid 2, and so on and so forth. So a messenger RNA is really an imprint or Xerox of the triplets in which order they are occurring on the gene. And so a messenger RNA is a copy of gene, right? It's a copy of the gene and specific order of amino acid or primary protein structure. So steps in protein synthesis, you have transcription, which occurs in the nucleus. It forms a messenger RNA. Messenger RNA moves into the cytoplasm, and you have translation of the messenger RNA by ribosomes and transfer RNA molecules to build the protein structure. So translation is the building of the protein structure in the cytoplasm. Transcribed in the nucleus, translated in the cytoplasm, which is the building of the reading of the messenger RNA and actually assembling the primary structure of the protein. Again, understand what a triplet is, understand what a codon is. Now, each codon codes for one amino acid, but one amino acid can have several codons that call for that amino acid. In addition, there's always an initiation codon, which says this is where the protein starts, and a termination codon, which says we are done building this protein. So both transcription and translation knows when to start and stop. There are 3 types of RNA transcribed, and RNA polymerase, which is the enzyme that oversees this synthesis of messenger RNA off a gene on DNA. It helps unwind the DNA template, and it helps add the codons to form the messenger RNA. And then it basically releases or when it reaches the termination and forms the messenger RNA. Messenger RNA, again, has the instructions for which amino acids need to go next to one another to form a primary structure of that particular protein. Ribosomal RNAs are actually the structures that form around the messenger RNA to read the messenger RNA codons and then tell the transfer RNAs to go find that specific amino acid and bring to the ribosomal complex to add to the protein strand. So transcription, there is the RNA polymerase that unwinds the DNA strand on the template strand of the gene. It also helps assemble the messenger RNA, which is assembling all these codons next to one another, and that which then becomes the messenger RNA. So here we go. Again, understand transcription and understand the definition of transcription and understand the definition of translation. Now, this podcast that we listened to in the last lecture talked about transcription factors, methylation. These switch on and turn on transcribing of proteins. So these methylations are what are initiating this RNA polymerase to start forming a messenger RNA to then go out and build a protein. And so again, if you want to listen to that podcast, it makes a little bit more sense when she starts talking about methylation and transcription factors and building proteins. And the proteins she's really talking about are those metabolic enzymes that help, move energy from the bonds of the food you ate to ATP molecules, so you can efficiently produce all these ATP molecules and keep going and use the energy that you have consumed. So at this point, you can kind of stop and read through initiation of a messenger RNA, how it gets elongated off the DNA strand and how it terminates, and go through these various next couple of slides of initiation, elongation, and termination. You can pick back up after transcription of gene. You can read through these. I want you to spend a second kind of going over this, and then start back up when you're ready to translation. So translation, just to show you, each one of these are amino acid. Leucine. You can see leucine is an amino acid that is really important and utilized in many, many different proteins. It has 6 different codons that code for that particular amino acid. Other amino acids like tyrosine only has 2, right? And valine has 4. So different amino acids have different numbers of codons that call for that particular amino acid, and that's just an FYI. But what's more important is once that messenger RNA leaves, this messenger RNA right here is going to leave through a nuclear pore, and it's going to be here. So this is the messenger RNA. That messenger RNA has a ribosome attached on top and on bottom, and this ribosome starts to read the codons. So the ribosome reads codons on the messenger RNA and then tells the transfer RNA what amino acids to go get. And that transfer RNA will go get the particular amino acids, bring it back to the ribosomal complex, and add that amino acid to the building chain of amino acids, which would be the polypeptide chain, right? So a number of amino acids. And that becomes the primary protein structure, which is the specific order of amino acids in that protein structure. These transfer RNAs, they read the codon. So transfer RNA reads codon on the messenger RNA and forms an anticodon for amino acid. It then goes out into the cytoplasm, finds the amino acid, binds it, and brings it to the messenger RNA ribosome complex to add to protein structure being built. So these tRNAs or transfer RNAs, if they have a anticodon, it's called charged, and it has an amino acid. If it doesn't have an anticodon on an amino acid, it is a free tRNA. And so this little slide here is kind of showing you, this would be, right, amino acid 1, this is amino acid 2, here is the codon on the messenger RNA, and it's doing an anticodon, which went and got that amino acid, brought it to the ribosomal complex, brought the second amino acid to the ribosomal complex, forms a peptide bond between them, and then this tRNA takes off. The next codon is read, the next tRNA is charged, and that amino acid is brought to the ribosomal complex, peptide bond is formed, and it's added, and so forth. And so it just keeps happening all the way down the messenger RNA till it hits a stop codon. The destination of these proteins is determined by what's called a leader sequence, and that leader sequence will tell the ribosome whether it needs to go to the rough endoplasmic reticulum, whether it stays in the cytosol, whether it gets packaged in a vesicle. If there's no leader sequence, it just can be a structural protein in this cell itself. So this is kind of the leader sequences and explanation, right? This could be a metabolic enzyme, right? This could be, you know, some kind of enzymes to break down bacteria or virus. And so it could be different things. Copying the DNA. So replication happens during mitosis. And you want to have an exact replication of the DNA because if something goes wrong, a UV ray hits it, some chemicals, the DNA doesn't get replicated perfectly. There could be an amino acid that is wrong in that gene. And that can make a protein be built that is functionally not correct, right? Because the structure of the protein determines the function of that protein. And if the correct amino acids aren't assembled in the correct order, it's not going to fold the right way. And ultimately the structure of that protein, the tertiary structure, is going to be wrong. And then the protein's not going to do its job correctly. So when this happens, it's called a mutation. It's where there is some change in the triplet and the DNA strand, and it's going to code for the wrong amino acid. And so the structure of the protein is going to change. And sometimes it's not a big deal. Sometimes it is a big deal. But that's what we call natural selection. Mitosis, by the way, is creating new cells from existing cells. Meiosis is what happens in the gametes. So you're having the number of chromosomes, and these would become gametes. So spermatozoa or oocytes. So we don't talk about that to the end. But that concludes today's lecture, and I look forward to seeing you soon.